Tonight, we're going to drift into one of the quiet foundations of life itself, the gentle world of DNA. This is Science for Sleep, a quiet place to unwind, rest, and drift through the softest corners of the universe at your own pace. Even though DNA is often described as a blueprint or a code, it behaves far less like a rigid instruction manual and far more like a soft, flowing record of who we are. Every strand carries stories reaching back through time, linking us quietly to our ancestors and guiding the delicate rhythms of our bodies. And yet, despite how familiar the word DNA has become, the true nature of this molecule remains remarkably subtle, filled with patterns, mysteries, and soft biological processes unfolding in near silence. For all the scientific progress we've made, there is still so much about DNA that is calm and unknowable. Entire sections of it remain unexpressed, resting like quiet pages in a book that the body rarely reads. Other segments flicker on or off depending on our environment, our experiences, and even the gentle ebb and flow of daily life. DNA hums underneath everything we do, but it does so with a patience and softness that makes it the perfect topic for a peaceful night's exploration. If you enjoy these slow, soothing journeys where curiosity meets calmness, I invite you to settle in, relax, and allow your mind to wander freely. There's nothing you need to remember, nothing you need to focus on. Just let each idea drift by like a quiet breath, easing you deeper into rest. Tonight, we'll explore DNA the same way we explore the night sky in our sleepy science videos, slowly, gently, and without any need for urgency. We'll uncover how DNA stores information, how it folds itself into shapes that are impossibly small, yet perfectly organized, how it guides the growth of every living thing, and how its patterns stretch across nearly all of life on Earth. We'll look at surprising quirks, unusual discoveries, and calm biological processes that continue whether we're thinking about them or not. You'll hear about how DNA repairs itself, how it unwinds and rewinds with incredible rhythm, how it protects the integrity of living systems, and how even the smallest changes can ripple softly across generations. We'll explore how DNA interacts with proteins, how it coils into elegant structures, how it mirrors itself, and how it carries instructions that are both intricate and astonishingly peaceful. This is a journey through the fabric of life, not in a fast, technical way, but in a quiet, relaxing one. A way that lets you appreciate the simplicity behind the complexity, the softness behind the science, and the calm order that lives beneath every cell of every living creature. So wherever you are, lying down, settling into bed, or simply looking for something gentle to unwind your thoughts, allow your breathing to slow, let your muscles soften, let the day grow quiet. There is nothing you need to do but listen. Tonight, we're not trying to understand everything. We're simply wandering through ideas, soft, sleepy facts about DNA that invite your mind to drift, settle, and rest. When you're ready, we'll begin our first section, drifting into the calming structure and surprising elegance of the molecule that makes life possible. DNA begins with a shape so simple and so elegant that it almost feels like something nature whispered into existence. At its core, DNA is a long molecule formed from two strands, gently twisted together into the familiar double helix. The strands wind around each other in a slow and graceful spiral, like two ribbons drifting in a calm breeze. Nothing about the structure is rushed. Every turn of the helix is steady, repeating itself again and again along the entire length of the molecule. Each strand is made of tiny chemical units called nucleotides. These nucleotides are the quiet alphabet of life, repeating themselves in long, delicate sequences. They come in 4 types: adenine, thymine, cytosine, and guanine. But in the peaceful world of DNA, they are known more simply as A, T, C, and G. These letters do not form words in the human sense, yet they carry meaning far deeper than any spoken language. They align themselves with calm precision, creating patterns that guide the growth, repair, and function of living organisms. The 2 strands of DNA are held together by gentle chemical bonds, the kind that form and break with ease, but remain stable under ordinary circumstances. These bonds pair A with T and C with G, creating soft connections across the helix. Each pair acts like a tiny bridge, linking the 2 strands together in perfect symmetry. The quiet predictability of these pairings is what allows DNA to copy itself so gracefully, with one sequence guiding the creation of its mirror image. Although the double helix looks small and delicate, it is surprisingly long. If you gently stretched out all the DNA in a single human cell, it would reach a length of nearly 2 meters. Yet all of this material is folded and wrapped so precisely that it fits inside a microscopic nucleus. The calmness of this arrangement is remarkable. DNA coils around proteins called histones, forming loops and folds that stack neatly into higher structures. This organization is so efficient that it maintains order even while allowing access to any part of the sequence whenever the cell needs it. The folding of DNA is not random. It follows patterns shaped by evolution, with regions that remain tightly packed and silent, and regions that stay more open, allowing the cell to read the instructions stored there. This dynamic arrangement shifts throughout the lifetime of the cell, slowly adjusting to internal signals, external conditions, and changes in the organism's environment. Even this process unfolds gently. DNA does not snap open or shut. It breathes, expanding and contracting in small, rhythmic motions that help maintain balance and stability. One of the quietest truths about DNA is that all of life on Earth shares its basic structure. Whether in a towering redwood tree, a tiny bacterium, or a person falling asleep to this video, the same double helix carries the instructions for existence. The same 4 letters appear in almost all living things, arranged in different sequences but following the same chemical rules. This unity reflects a deep connection across the tree of life, a shared molecular ancestry that links every organism to the earliest forms of life that appeared on our planet billions of years ago. Despite its simplicity, DNA behaves with surprising flexibility. It bends and twists without breaking, adjusting to the needs of the cell. In some cases, it forms unusual shapes, loops, knots, and even 4-stranded structures that appear only under certain conditions. These shapes influence how genes are read, how cells respond to stress, and how quickly DNA can make copies of itself. Yet even when forming these VD variations, the molecule retains its underlying calmness. The changes occur without drama, guided by soft chemical forces that help maintain stability. DNA also has regions known as promoters and enhancers, quiet signals that help the cell determine where, when, and how strongly a gene should be activated. These regions don't code for proteins themselves, but they influence the rhythm of gene expression. Some genes are expressed constantly, providing essential instructions that the cell needs at all times. Others activate only under certain conditions, turning on gently and turning off just as quietly. This balance allows organisms to adapt while still maintaining order. Another remarkable aspect of DNA is its symmetry. Each strand contains information that complements the other. If one strand is damaged, the cell can often repair it using the unbroken strand as a reference. This self-correcting property helps preserve the integrity of genetic information across generations. Even when errors occur, as they inevitably do, the repair systems work patiently, scanning for mismatches and restoring the proper sequence. In many ways, DNA is less like a mechanical instruction manual and more like a living, breathing library. Some sections are read frequently, others only on rare occasions, and many remain untouched for long periods. Yet all sections are preserved with care, folded neatly within the nucleus. This library does not operate on urgency, it operates on rhythm. Chemical signals guide the cell to the pages it needs, while other regions rest in quiet darkness, waiting for a moment when their instructions might be required. When you think about DNA on a cosmic timescale, its calmness becomes even more profound. Across billions of years, life has changed, adapted, and evolved, yet the structure of DNA has remained remarkably consistent. The same helical pattern that winds through your cells also wound through the cells of ancient organisms that lived long before humans existed. And long after we are gone, life on Earth, whatever form it takes, will likely still rely on the gentle spiral of DNA. As we drift deeper into the quiet world of genetics, we begin to see DNA not just as a molecule, but as a peaceful framework that supports life with subtle efficiency. Its structure, simple, elegant, and enduring, serves as the foundation for everything that follows. DNA stores information in a way that is both incredibly simple and deeply profound. Every instruction that guides life is stored in long sequences of the 4 familiar bases, A, T, C, and G. These letters combine in different patterns, creating gentle chemical sequences that the cell can read and interpret. Even though each individual base is small, together they form a quiet language capable of describing the entire structure of a living organism. What makes DNA especially peaceful is its consistency. No matter what type of organism you look at, the language is the same. A pairs with T and C pairs with G. This pairing is predictable, stable, and calm. The cell relies on these relationships to copy and preserve information. Every time the DNA replicates, it uses these pairings to recreate the correct sequence, allowing life to continue with remarkable fidelity. Within the long stretches of DNA, certain regions hold the instructions for making proteins. These regions are called genes. Each gene contains a sequence that the cell can translate into a specific protein. Proteins perform countless functions, building structures, regulating processes, and supporting the quiet routines that keep cells alive. Yet DNA does not force the cell to read every gene at once. Instead, it keeps information tucked away until the right moment. The order of the bases in each gene determines the structure of the protein it encodes. A small change in that order can lead to a different protein, or sometimes no protein at all. Yet most of the time, DNA remains steady and unchanged, allowing cells to function with reliable harmony. When changes do occur, they often arise slowly, passing from one generation to the next. These gentle variations are part of how life evolves over long stretches of time. But DNA doesn't just store protein coding information. Much of the DNA in an organism is non-coding. For many years, scientists didn't understand what purpose these regions served. They seemed silent, like long, empty pages in a book. Today, we know that many of these regions play subtle roles in regulating how genes behave. Some act as switches that turn genes on or off. Others help shape the structure of the DNA, influencing how accessible certain regions are. Because of this, DNA is sometimes compared to a vast library. Only a small portion of the books contain detailed instructions for building proteins. The rest hold information about how to organize those books, how to retrieve them, and how to maintain the library's internal order. Even if these sections appear quiet or unused, they contribute to the gentle orchestration of cellular life. DNA also stores information in the way it folds. The physical shape of the DNA molecule influences which genes are active at any moment. When a region is tightly coiled, the cell may not be able to read it easily. When the region relaxes, the instructions become accessible. This dynamic folding creates a soft interplay between structure and function, a relationship that helps the cell respond calmly to its environment. The information in DNA is remarkably compact. A single gram of DNA could theoretically store more data than any modern computer system by orders of magnitude. This density reflects the quiet efficiency of the molecule. Rather than spreading its information out across large structures, DNA condenses everything into a form so small that billions of copies can coexist in a living organism. Another gentle feature of DNA's information storage is redundancy. Certain processes in the cell read multiple bases at a time, forming codons, small three-letter units. Many codons correspond to the same amino acid, meaning the cell has multiple ways to construct the same protein. This built-in redundancy helps protect the system from small errors. If a single base changes, the overall meaning might still remain intact. In addition to the nuclear DNA housed within the cell's nucleus, many organisms also contain mitochondrial DNA. Mitochondria, often described as the cell's energy centers, carry their own small loops of DNA. These loops store information essential to energy production. They are inherited through the maternal line, forming quiet genetic threads that pass from generation to generation with gentle continuity. The information stored in DNA also reflects the history of life. Hidden within the sequences are traces of ancient events, past infections, evolutionary changes, and old genetic codes that have been carried along through time. Some sequences appear repetitive or seemingly inactive, but they represent remnants of long-forgotten biological stories. These quiet echoes remain embedded in the genetic material, linking modern organisms to the deep past. Even though DNA stores so much information, the cell never reads all of it at once. Different genes activate in different types of cells. A neuron reads a different set of instructions than a muscle cell. A skin cell reads a different set than a blood cell. This selective reading allows a single genetic blueprint to create many specialized forms, each working comfortably within its own role. DNA's ability to store information with such accuracy and consistency is one of the reasons life can be so stable. Yet its ability to adapt through small changes in sequence or expression is what allows life to be flexible. This balance, steady but not rigid, is part of what makes DNA such a calming topic to explore. The molecule does not rush to change. It shifts slowly, guided by long-term pressures and soft chemical influences. As we drift deeper into the quiet world of genetics, the way DNA stores information becomes even more remarkable. It is not simply a code. It is a collection of possibilities, a gentle framework that supports growth, diversity, and continuity. Every organism carries its own version of this information, shaped by countless generations that came before. DNA replication is one of the quietest and most elegant processes inside a living cell. It happens constantly, peacefully, and with a rhythm that feels almost like breathing. Every time a cell prepares to divide, it must make a complete copy of its DNA, so that each new cell receives the full set of instructions. Even though this process is incredibly complex, it unfolds with a calm precision that makes it one of the most soothing topics in biology. Replication begins with the unwinding of the double helix. At first glance, the tightly coiled DNA seems like it would resist being opened, but the molecule is built for flexibility. Special proteins gently attach to specific regions of the DNA and begin to separate the two strands, creating a small opening known as a replication fork. This opening is not sudden or dramatic. It expands slowly, like the first pages of a book being opened to reveal the story inside. Once the strands separate, each one serves as a quiet template for building a new complementary strand. This complementary relationship, a pairing with T and C pairing with G, is what allows the cell to replicate its DNA with such remarkable accuracy. The original strands act like guides, ensuring that the new strands are created in perfect alignment. The enzyme responsible for building the new strands is called DNA polymerase. Despite its complicated name, its behavior is surprisingly gentle. DNA polymerase moves slowly along the exposed strand, reading each base and selecting the appropriate match. It adds one nucleotide at a time, forming new bonds with soft consistency. This enzyme works with such care that it often corrects mistakes immediately, removing mismatched bases and replacing them with the correct ones. Its patience contributes to the stability of genetic information across generations. Replication happens in two directions. One new strand is built continuously as the polymerase follows the unwinding movement of the helix. This strand is called the leading strand. The other, known as the lagging strand, is built in small fragments. Even though this process sounds more complicated, it still unfolds with a slow, careful rhythm. The fragments are eventually connected by another enzyme, forming a smooth, continuous strand. Throughout replication, many helper proteins support the process. Some stabilize the separated strands to prevent them from winding back together. Others relieve the tension created by the unwinding motion. Each protein works quietly, contributing to a seamless operation that feels almost orchestral. Every molecule plays its part, and together they create a peaceful environment where the DNA can be duplicated with precision. The replication forks move steadily along the molecule until the entire DNA sequence has been copied. This can take hours in some cells, but the pace is deliberate and calm. The cell is in no rush. Its priority is accuracy, not speed. The slow, rhythmic activity of the replication machinery ensures that the new strands match the originals as closely as possible. Once replication is complete, the cell gently checks the newly created DNA for errors. Repair enzymes scan the strands, looking for mismatches or distortions. If they find something unusual, they make quiet corrections. This proofreading ensures that the genetic information remains steady and reliable, even across countless cell divisions. One of the most beautiful aspects of DNA replication is its symmetry. Each new DNA molecule contains one strand from the original double helix and one newly synthesized strand. This creates a peaceful continuity between generations of cells, a gentle preservation of biological memory. The process is known as semi-conservative replication, reflecting the balance between old and new. Replication also plays a role in protecting the ends of DNA molecules. At the tips of each chromosome lie structures called telomeres. These regions do not code for proteins. Instead, they serve as buffers, preventing essential information from being lost during replication. As cells divide repeatedly, telomeres gradually shorten. This slow shortening is connected to aging, but even this process is gentle and gradual, unfolding over many cycles. Some cells, such as stem cells and certain immune cells, possess an enzyme called telomerase. This enzyme helps restore telomere length, allowing these cells to replicate more times than ordinary ones. Telomerase operates with quiet steadiness, adding small segments to the ends of chromosomes and helping preserve the cell's ability to divide. The calm efficiency of DNA replication is one of the reasons why life can grow and replenish itself. Skin cells, blood cells, and countless other tissues rely on replication to maintain their populations. Without this gentle copying process, organisms would not be able to heal injuries, grow new cells, or sustain themselves. Even though the replication machinery involves many moving parts, the overall process feels remarkably serene. It is a coordinated dance of molecules unfolding at a pace that supports stability in life. Every step, from the unwinding of the double helix to the final proofreading, takes place in silence, hidden within the microscopic world of the cell. As we drift deeper into the quiet mechanics of DNA, replication stands out as one of the most peaceful demonstrations of nature's patience. It is not rushed. It is not forceful. It is a steady, dependable rhythm that has carried life forward for billions of years. Inside every cell, DNA does not work alone. Its calm structure is supported, protected, and guided by an entire community of proteins. These proteins read its instructions, fold it into gentle shapes, repair its strands, and help regulate when certain genes are expressed. Together, DNA and proteins form one of the most peaceful partnerships in biology, a quiet collaboration that keeps life balanced and functioning. One of the first protein partners that DNA meets is the histone. Histones are small, rounded proteins that act like spools, allowing DNA to wrap neatly around them. Because DNA is long and delicate, it cannot remain loose inside the nucleus. Instead, it coils gently around histones, forming soft loops known as nucleosomes. These loops help keep the DNA organized, preventing it from becoming tangled or exposed. The wrapping of DNA around histones is not just for storage. It also influences gene expression. When DNA is tightly coiled around these proteins, the cell cannot easily access the underlying sequence. When the coils loosen, certain genes become more available for reading. This gentle shifting between open and closed states helps the cell adapt without sudden changes. The motion is quiet and rhythmic, a slow adjustment to internal signals or environmental cues. Other proteins, known as transcription factors, play a role in determining which genes should be active at any given moment. These proteins bind to specific regions of the DNA, guiding the cell's machinery to the correct starting point. Their interactions are delicate. A transcription factor does not force DNA to open. It simply rests on the helix, signaling where the cell should pay attention. This subtle guidance helps maintain the harmony of gene expression. When a gene needs to be read, an enzyme called RNA polymerase gently attaches to the DNA. This enzyme moves along the strand, forming a temporary transcription bubble, a slight separation of the two DNA strands. As RNA polymerase travels, it constructs a complementary RNA molecule that mirrors the gene sequence. This RNA carries the instructions needed to build a protein. The entire process unfolds slowly and steadily, with each step controlled by the calm chemistry of the cell. After the RNA is created, it interacts with the ribosomes, large protein RNA complexes responsible for assembling amino acids into proteins. Ribosomes move along the RNA like quiet readers following a line of text. Each three-letter codon in the RNA corresponds to a specific amino acid. As the ribosome progresses, it gently adds each amino acid to the growing protein chain. There is no hurry. The rhythm is consistent, shaping each protein with patient precision. Chaperone proteins assist during this stage. They help newly formed proteins fold into their proper shapes. Folding is essential because a protein's shape determines its function. Chaperones act like guides, ensuring that the folding process remains smooth and controlled. They do not impose shape with force. Instead, they create a protected environment where the protein can settle into its natural structure. Even in this intricate process, the overall feeling is one of calm and gentle support. Some proteins work closely with DNA to repair damage. Life inside a cell is dynamic, and DNA can occasionally be affected by radiation, chemicals, or natural errors. During replication, repair proteins slowly travel along the DNA, scanning for irregularities. When they detect a mismatch or distortion, they correct it by removing the damaged segment and replacing it with the proper sequence. These repairs happen continuously, preserving the integrity of the genetic material with quiet vigilance. There are also proteins responsible for regulating the movement and organization of DNA during cell division. Before a cell divides, the DNA condenses into chromosomes, compact structures formed by wrapping and folding the DNA more tightly than usual. Structural proteins help guide this process, ensuring that each chromosome is shaped correctly. When the cell is ready to split, other proteins help gently pull the duplicated chromosomes apart, delivering them to opposite sides of the cell with steady coordination. Another remarkable protein complex involved in DNA's life is the replisome, a collection of enzymes and helper proteins that assist with DNA replication. DNA polymerase is part of this complex, but many other proteins contribute as well. Some unwind the helix, others stabilize the strands, and others join DNA fragments together. Each protein performs its role in harmony with the others, creating an environment where replication can unfold smoothly. Proteins also help manage the physical tension that DNA experiences. As the molecule unwinds and rewinds, it can become twisted or strained. Topoisomerases are the proteins responsible for relieving this tension. They work by temporarily cutting one or both strands of the DNA to allow it to relax before resealing the break. Even though this sounds dramatic, the process is extremely controlled. Topoisomerases perform these actions with precision, maintaining the DNA stability while gently releasing stress. Inside mitochondria, similar protein interactions occur with mitochondrial DNA. Because mitochondrial DNA is circular and organized differently, the proteins that maintain it are specialized for that environment. These proteins assist with replication, repair, and transcription in a way that mirrors the roles of nuclear proteins, but reflects the unique conditions inside the mitochondria. The partnership between DNA and proteins is a relationship built on balance. DNA provides the instructions and proteins carry them out. Yet DNA also relies on proteins for protection, organization, and repair. This cooperation ensures that the cell remains stable, even as countless processes occur within it every moment. When you look closely at this molecular partnership, you begin to appreciate how quietly life operates. Each protein interacts with DNA at the right time, in the right place, and with the right gentleness. Together, they create a system capable of supporting the growth and evolution of every organism on Earth. Even though all humans share the same basic structure of DNA, the same double helix the same 4 chemical bases, and the same gentle pairing rules. The sequences that make each person unique vary in soft and subtle ways. These differences form the quiet diversity that shapes everything from our appearance to the way our cells respond to the world around us. And yet, despite this variation, the similarities between any two people far outweigh the differences. In fact, human DNA is more than 99.9% identical across the entire species. This means that only a tiny fraction, less than 0.1%, contributes to the unique genetic features of each individual. These differences are not dramatic. They are small, gentle changes in the arrangement of bases along the DNA. A slight shift here, a quiet substitution there, and the pattern becomes distinct. But because DNA sequences are so long, even tiny differences can influence the characteristics we carry. One of the most common sources of variation is called a single nucleotide polymorphism, often shortened to SNP. A SNP is a change in just one letter of the DNA sequence. Instead of an A, there may be a G. Instead of a C, there may be a T. These small shifts occur naturally over generations. Most have no noticeable effect on the person who carries them. They simply contribute to the quiet tapestry of human diversity. Some SNPs influence visible traits, such as hair color, eye color, or certain aspects of facial structure. Others affect how our bodies process nutrients, respond to environmental factors, or interact with medications. But most exist without any obvious consequence. They drift through populations gently, passed from parents to children, creating subtle patterns that reflect ancient migrations and shared ancestry. Another form of variation arises from differences in the number of times certain sequences repeat. These repeating regions are known as microsatellites or short tandem repeats. They occur throughout the genome, like calm, rhythmic patterns in the DNA. In some people, a particular sequence might repeat 10 times. In others, it might repeat 12. These differences are used in genetic studies to trace family relationships or understand the movement of populations across time. But within the body, they are simply quiet variations that sit comfortably within the larger structure of the genome. Human DNA also contains segments known as haplotypes, clusters of SNPs that tend to be inherited together. These haplotypes form gentle patterns across populations, reflecting distant histories and shared lineages. They help researchers understand how groups of people are connected across continents and generations. But for the individuals who carry them, haplotypes are simply part of their genetic identity, resting quietly within their cells. Even though each person has a unique set of genetic variations, the impact of these differences is only part of the story. Many traits arise from a combination of genetic influences and environmental experiences. Genetics provides the foundation, but lifestyle, nutrition, surroundings, and even chance shape how those genes are expressed. In this way, DNA creates a framework, and life gently fills in the details. Some variations in DNA play roles in health. Certain genetic differences may increase or decrease the likelihood of developing particular conditions. But even here, the relationship is often complex and gradual. Most conditions arise from the interplay of many genes, each contributing a small influence. It is rare for a single variation to determine a trait entirely. Instead, the effects accumulate softly, shaped by interactions among genes and environmental cues. There are also variations that arise spontaneously, small changes that occur when DNA is copied during cell division. These changes, known as mutations, appear quietly and usually remain benign. Many occur in regions of DNA that do not affect gene function. Others are repaired by the cell's diligent repair systems before they can have any effect. Only a tiny fraction of these variations lead to noticeable differences, and even then, the effects are often subtle. Across the world, human DNA reflects a long history of movement, adaptation, and shared origins. Populations carry variations that trace their ancestry to different regions, climates, and ancient events. Yet the deeper message remains the same. All humans are connected through a broad, gentle web of genetic similarity. The differences that do exist are small threads woven into the larger pattern of our shared biology. Even identical twins, who share nearly the same DNA, develop subtle differences over time. Environmental influences, epigenetic changes, and personal experiences shape how their genes are expressed. These differences unfold slowly across a lifetime, reflecting the adaptability of the genome. It shows that DNA is not a static script, but a dynamic, responsive blueprint that shifts gently in response to life's rhythms. As we rest with these ideas, it becomes clear that diversity in DNA is not loud or abrupt. It is soft, gradual, and deeply natural. The quiet differences that make each person unique arise from calm variations in sequence, expression, and environment. They help shape the world's rich tapestry of human experience, while reminding us of our shared origins. From the moment life begins, DNA becomes the quiet guide that shapes every stage of development. Even before a person is aware of the world, before the first breath or the first heartbeat, DNA is already at work, softly directing the formation of cells, tissues, and organs. Its instructions unfold slowly and patiently, ensuring that each step in the early development of life occurs with delicate coordination. At the earliest stage, after fertilization, a tiny single cell carries the full complement of DNA needed to build an entire human body. This single cell contains two sets of genetic instructions, one from each parent, that merge to form a unique and complete blueprint. Yet the cell does not read the entire blueprint at once. Instead, it begins with the simplest task, dividing. As the first few cell divisions occur, the DNA is replicated gently and repeatedly, ensuring that each new cell receives an identical set of instructions. These early cells are called totipotent cells, meaning they have the potential to become any cell type in the body. They hold all possibilities within them, guided by the quiet presence of DNA. As development continues, these cells begin to specialize. Some will form structures that support growth, while others will begin the transformation toward becoming specific tissues. The DNA inside each cell remains the same, but the way the cell interprets the DNA becomes more focused. This is part of the beautiful paradox of development. Identical genetic material leads to different outcomes based on how it is read. One of the earliest and most important processes is called differentiation. Differentiation is the gradual process in which cells adopt specific roles. In one region of the embryo, cells begin to express genes related to muscle formation. In another, cells activate genes that will guide the creation of the nervous system. These shifts happen slowly, with DNA acting like a calm conductor guiding an orchestra of molecular signals. During this time, chemical cues within the developing embryo help determine which genes should be active. Proteins and signaling molecules move softly through the cellular environment, attaching to different regions of the DNA to encourage or silence certain instructions. This selective reading creates the foundation for the body's major structures. Some cells will become neurons, others will become bone, others will become skin or blood, or the delicate tissues of the heart and lungs. The instructions for all of these outcomes lie quietly within the DNA, waiting for the right moment to be expressed. As the embryo continues to grow, DNA also guides the formation of the body's major axes: the top and bottom, the front and back, the left and right. This organization helps ensure that structures appear in the correct places. Genes known as homeobox genes, or HOX genes, play a key role in this patterning. They act like gentle markers along the body's blueprint, telling cells where they are located and what they should become. Their sequences are deeply conserved across species, reflecting their essential role in development. Another important aspect of early development is the creation of the germ layers. These layers, the ectoderm, mesoderm, and endoderm, form the foundation for all of the body's tissues. The ectoderm gives rise to the skin and nervous system. The mesoderm forms muscle, bone, and the circulatory system. The endoderm becomes the digestive and respiratory systems. Each layer contains cells that read the same DNA, but interpret it in ways that create unique structures. During these early stages, epigenetic changes begin shaping which genes remain active and which remain silent. Epigenetic marks, such as DNA methylation or histone modification, gently influence gene expression without altering the underlying DNA sequence. These marks help lock in certain patterns of development, ensuring that once a cell commits to a particular role, it maintains its identity. For example, a muscle cell will continue to express muscle-related genes throughout its life, even though it carries the instructions for all other cell types. This stability gives the developing body structure and order. The calm interplay between DNA and epigenetic regulation creates a foundation that supports the organism throughout its lifetime. DNA also guides the formation of the nervous system, beginning with a simple neural plate. This flat sheet of cells gradually folds into a neural tube, the precursor to the brain and spinal cord. As the neural tube develops, neurons begin to form, each following gentle molecular cues that guide their growth and connections. The complexity of the nervous system emerges from countless small instructions, each softly encoded within the DNA. As the heart forms, DNA directs the timing and pattern of cell movement and tissue formation. The heart begins as a simple tube, beating softly even in early development. Over time, the tube loops and folds, creating chambers and valves. These changes occur with quiet coordination, driven by gene networks that operate like a peaceful sequence of steps. Throughout development, the entire process remains deeply efficient. DNA offers guidance, but the cell's environment, nutrients, signals, temperature, and timing also plays a role. This interplay between genetics and surroundings creates an adaptable, resilient system that can adjust gently to changes. By the time a developing organism is ready to enter the world, trillions of cells have formed, each carrying identical DNA, yet each fulfilling a distinct role. The instructions woven into the genome have guided the creation of organs, systems, and structures, all arising from a single fertilized cell. This slow, steady unfolding of life is one of the most peaceful processes in nature. DNA provides the blueprint, but it does so without force. Its instructions exist quietly, waiting for the right signals and the right conditions to bring each step into motion. Inside each cell, DNA serves as a quiet library of instructions, but it is the process of gene expression that determines which pages are read and when. Not all genes are active at the same time. Some remain silent for long stretches, resting peacefully in tightly coiled regions of the genome. Others are gently opened when the cell needs their guidance, allowing the information inside to flow into the machinery that shapes life's quiet rhythms. Gene expression begins with transcription, the calm process in which a segment of DNA is copied into RNA. This copying is not abrupt. It starts with the gradual binding of proteins that recognize specific sequences along the DNA. These proteins act like soft markers, letting the cell know where a gene begins. Once in place, they help open the DNA slightly, creating a small space where the instructions can be accessed. RNA polymerase then settles onto the exposed DNA. It moves slowly along the strand, reading each base and forming a matching RNA molecule in response. This RNA is a temporary messenger, carrying the gene's information out of the nucleus and into the cytoplasm, where proteins are built. The pace of transcription reflects the cell's needs. Some genes are transcribed continuously, providing essential components for everyday functions. Others activate only under specific conditions, responding gently to signals such as temperature changes, nutrients, or subtle shifts in the environment. After the RNA is created, it undergoes a quiet refining process. Some segments of the initial transcript, called introns, are removed. The remaining segments, known as exons, are joined together to create a smooth, continuous message. This process allows the cell to shape a single gene into different versions of a protein by choosing which exons to include. The calm flexibility of this editing adds depth and variety to the instructions encoded in the DNA. When the RNA is ready, it travels through small pores in the nuclear envelope and enters the cytoplasm. There, ribosomes read the message slowly and steadily. Each three-letter codon corresponds to an amino acid, and the ribosome assembles these amino acids one by one into a growing chain. The chain folds gently into a functional protein, guided by the chemical properties of its components and by helper proteins that ensure a smooth process. Every step unfolds with a quiet, deliberate rhythm. Gene expression is influenced by many factors. Some genes respond to internal signals that reflect the cell's condition. Others react to external cues, such as light, temperature, or the presence of nutrients. This responsiveness allows organisms to adapt gracefully to their surroundings. Even though the DNA sequence remains stable, the way it is interpreted can shift subtly during different stages of life or in response to gentle environmental pressures. Epigenetic marks help regulate gene expression. These marks modify the accessibility of DNA without altering the underlying sequence. One such mark, DNA methylation, involves the addition of a small chemical group to specific bases. When a region is heavily methylated, it becomes less accessible, and its genes are less likely to be expressed. Histone modifications also play a role. They can tighten or loosen the DNA's coils, influencing how easily the cell can read the associated genes. These epigenetic patterns reflect the cell's experiences. Over time, nutrition, stress levels, physical activity, and even patterns of rest can leave soft imprints on the genome. While some marks fade, others may persist, guiding gene expression in subtle ways. This adds a layer of plasticity to the genome, allowing organisms to adjust quietly to their environment across days, years, or generations. Gene expression also follows circadian rhythms. Certain genes activate more strongly during the day, while others rise gently at night. These daily cycles help regulate sleep, digestion, hormone levels, and countless other processes. The shifting pattern resembles a calm ebb and flow, aligning internal activities with the timing of the outside world. In multicellular organisms, gene expression varies across tissues. Liver cells express genes related to metabolism and detoxification. Neurons focus on genes involved in signaling and communication. Skin cells activate genes that support protection and repair. This specialization arises not from differences in DNA, but from differences in which genes are quietly brought to life. Sometimes, gene expression shifts as part of long-term adaptation. When an environment changes, a new climate, a new diet, a new source of stress, certain genes may be expressed more frequently, while others recede. These changes occur gradually, giving organisms time to adjust. The flexibility of gene expression provides a gentle buffer against the unpredictability of life. Throughout all these processes, the genome remains steady. It provides the instructions, but it does not insist on how they are used. Gene expression allows the cell to decide which instructions to follow in each moment, creating a dynamic but peaceful conversation between the DNA and the environment. Even though DNA is remarkably stable, it is not completely unchanging. From time to time, small variations appear within the sequence, gentle shifts known as mutations. These changes arise naturally, woven into the quiet processes of replication, repair, and environmental interaction. Most mutations are harmless. Many are silent, altering nothing at all. But whether they influence traits or remain unnoticed, they are part of the natural ebb and flow of life's genetic history. Mutations can take many forms. The simplest is a substitution, where one base is replaced by another. An A might become a G, or a C might shift to a T. These tiny changes often occur during replication, when DNA polymerase occasionally misreads a base. Even then, most mistakes are corrected immediately. Repair enzymes scan the DNA with calm vigilance, restoring the correct sequence. Only a small number of substitutions remain, drifting quietly through the genome. Another type of mutation is an insertion or deletion, where a base or a small group of bases is added or removed. These changes can alter the rhythm of the genetic code, shifting how the cell reads a sequence. Yet even here, many effects are subtle. Some regions of the genome tolerate variations more easily than others. Insertions and deletions sometimes occur in non-coding regions, where they settle without disrupting essential instructions. Larger mutations can also occur. Segments of DNA may be duplicated, creating extra copies of a gene or a portion of a gene. These duplications can introduce gentle flexibility into evolution. One copy may continue performing its original role, while the other is free to accumulate changes over generations. This process can lead to novel functions, adding depth to the complexity of life. Occasionally, sections of DNA may be rearranged. A portion might flip its orientation, or two distant segments might exchange places. These rearrangements are rare and generally occur over long periods of evolutionary time. For the organism, they often pass quietly, unnoticed and undisturbing, unless they happen to affect a sensitive region of the genome. Mutations can arise from the environment as well. Sunlight, for example, carries ultraviolet radiation that can subtly affect DNA. When UV light interacts with certain bases, it may cause them to bond in unusual ways. The cell responds by dispatching repair proteins that correct the distortion. This process is continuous, a quiet defense against the gentle pressures of the outside world. Sometimes chemicals can interact with DNA, altering bases or interfering with replication. The body's repair systems address many of these changes, restoring the proper sequence. Even when a mutation remains, it often causes no significant effect. The genome contains vast stretches of non-coding sequence, where small changes settle without influencing the organism's traits. Mutations also play a role in evolution. Over countless generations, they introduce subtle variation into populations. Most changes are neutral, neither helpful nor harmful. Some provide small advantages that help individuals adapt to their environment. These beneficial mutations may persist and spread slowly, shaping the gradual evolution of species. Neutral mutations are especially common. They move through populations quietly, carried from parent to child without influencing survival. Over long spans of time, these neutral changes accumulate, forming gentle signatures that help trace evolutionary relationships. Scientists can compare these sequences across species to reconstruct ancient lineages and understand how life diversifies. Harmful mutations are less common, but they exist. When a mutation disrupts an essential gene, it can affect the organism's well-being. However, most harmful mutations occur in only one copy of a gene, softened by the presence of a normal copy. The cell continues functioning, supported by redundancy within the genome. Only when mutations affect both copies do the effects become more pronounced. Some mutations occur in germ cells, the cells that produce eggs and sperm. These changes can be passed to the next generation, becoming part of a family's genetic history. Others occur in somatic cells, the cells of the body, where they affect only the individual. Somatic mutations arise as part of the normal wear and tear of life, accumulating slowly as cells divide and respond to their environment. Mitochondrial DNA has its own mutation patterns. Because mitochondria generate energy, they are exposed to reactive molecules that can influence their DNA. Even so, mitochondrial mutations accumulate gradually, often without causing noticeable effects. Like gentle strokes in a long painting, they contribute to the subtle variations that separate individuals and populations across time. One of the most peaceful truths about mutations is that they are natural. They are not signs of instability or failure. They are quiet reflections of the fact that life is dynamic and ever-changing. Every organism carries mutations, inherited from ancestors or acquired throughout life. These variations shape the diversity of nature and allow species to adapt to new conditions. Mutations are not sudden interruptions in the calmness of the genome. They are soft adjustments, occurring at a pace that aligns with the slow unfolding of evolution. Over millions of years, countless small changes accumulate, guiding the transformation of species, the emergence of new traits, and the branching paths of life's long history. Even within a single individual, the genome is not a static script. It shifts gently over time, responding to life's experiences and the natural processes of the cell. These quiet changes enrich the story written in DNA, adding new layers to the narrative passed from one generation to the next. Even though DNA is remarkably resilient, it faces constant challenges. Everyday activities, breathing, eating, moving, simply existing, expose the genome to small stresses, yet the cell responds with a calm, continuous effort to protect and restore its genetic material. DNA repair is one of the most peaceful, steady processes in biology, quietly preserving the integrity of life's essential instructions. Every moment, repair proteins drift through the nucleus like gentle caretakers. They scan the DNA for irregularities, moving slowly along the strands and pausing when something appears out of place. These proteins do not act with urgency. They operate with patience, recognizing mismatched bases, small breaks, or subtle distortions, and addressing each issue with careful precision. One of the simplest repair mechanisms is called base excision repair. When a single base is damaged, perhaps by a small chemical interaction or the natural wear of cellular processes, a repair enzyme removes the faulty base with soft efficiency. Another enzyme then inserts the correct base restoring the sequence. The entire process happens quietly, without disrupting the cell's overall calm. Another repair process, known as nucleotide excision repair, handles larger distortions. When UV light causes two bases to bond incorrectly, or when a bulky chemical attaches itself to the DNA, this pathway steps in. A small section of the damaged strand is gently cut out. Then, using the undamaged strand as a guide, the cell rebuilds the missing piece. The result is a restored and steady double helix, ready to continue its essential work. Mismatch repair is another layer of protection. During replication, DNA polymerase occasionally places the wrong base opposite a template. Most mistakes are corrected immediately, but if one slips through, mismatch repair proteins detect the inconsistency. They remove the mismatch region and allow the polymerase to try again. This system adds an extra level of calm accuracy to the copying of genetic information. Some forms of damage affect both strands of DNA. These double-strand breaks may arise from radiation, reactive molecules, or natural mechanical stress within the chromosome. When this occurs, the cell uses pathways such as homologous recombination or non-homologous end joining to reconnect the strands. Homologous recombination is especially gentle, using an intact copy of the sequence as a template to rebuild the damaged area. Non-homologous end joining is quicker but still controlled, reconnecting the strands in a way that preserves stability. Repair processes also help maintain the structure of telomeres at the ends of chromosomes. Telomeres protect valuable genetic information during replication, acting as soft buffers that absorb gradual shortening. Telomerase, the enzyme responsible for extending telomeres, works slowly and carefully, adding bases one at a time. Its activity helps certain cells continue dividing, supporting growth, renewal, and long-term function. In some cells, DNA damage may activate a gentle pause in the cell cycle, a moment of stillness where the cell stops dividing to give the repair mechanisms time to work. This pause helps prevent errors from being passed to new cells, maintaining stability through generations of division. Once the repair is complete, the cell resumes its rhythm, continuing its quiet routines. DNA repair is so effective that most damage remains unnoticed. Countless small corrections occur each day in every cell, preserving the sequence with remarkable consistency. Even when the damage cannot be fully repaired, the cell often adapts by silencing the affected region or activating redundant pathways. The genome's resilience reflects a balance between vulnerability and protection, created by billions of years of evolution. Across the lifespan of an organism, repair systems help protect against the gradual accumulation of mutations. While some variations remain, the majority of damage is corrected before it can influence the body. This steady maintenance supports long-term health, allowing cells to perform their functions reliably. Different tissues experience different levels of DNA repair. Cells exposed to sunlight, chemicals, or constant turnover may rely more heavily on repair pathways. Other cells, such as neurons, divide rarely and focus on preserving their DNA over long periods. Yet in all tissues, the underlying processes remain gentle and precise. As organisms age, repair mechanisms may slow. Telomeres shorten, repair proteins become less active, and small mutations accumulate gradually. These changes reflect the natural passage of time, a quiet softening of the genome's defenses. Even so, the resilience of DNA persists. Many cells continue to maintain their sequences with surprising accuracy late into life. Across all forms of life, from simple bacteria to complex animals, DNA repair remains a fundamental part of survival. It protects organisms from environmental challenges and internal stresses, ensuring that the genetic instructions remain steady through cycles of growth, division, and adaptation. The calmness of DNA repair reveals one of the most beautiful truths about biology. Even in a world full of change, life maintains its stability with gentle, continuous care. Each correction, each restoration, each quiet adjustment contributes to the long-term harmony of the genome. Although DNA is a long, delicate molecule, it does not float loosely inside the cell. Instead, it organizes itself into structures known as chromosomes, quiet, orderly arrangements that protect the genome and ensure its instructions are passed on with gentle precision. These structures help DNA remain stable through countless cycles of growth and division, serving as the architectural framework for genetic information. In humans, most cells contain 46 chromosomes, arranged in 23 pairs. Each pair consists of one chromosome inherited from the mother and one from the father. Even though these chromosomes hold immense amounts of information, they remain folded and compact, fitting comfortably within the microscopic nucleus. Their organization reflects billions of years of evolution, shaping the calm and efficient strategies that cells use to preserve life. For most of the cell's life, chromosomes remain in a relaxed, uncondensed state. During this time, the DNA is accessible, allowing the cell to read the instructions it needs. The strands loop and fold gently around histones, forming a quiet landscape of fibers known as chromatin. This landscape is dynamic, shifting in response to signals that activate or silence specific genes. In this open form, chromosomes behave like peaceful libraries, offering access to the pages required for growth and function. As the cell prepares to divide, the chromatin gradually condenses. The DNA coils more tightly, wrapping around itself in layers of increasing density. These layers form the familiar X-shaped structures seen in textbooks, though this shape appears only briefly during cell division. The condensation protects the DNA, preventing tangles or breaks as the cell separates its genetic material into two new cells. Each chromosome consists of two identical sister chromatids during division. These chromatids are connected by a region called the centromere, which serves as a quiet anchor point. The centromere ensures that each chromatid is pulled gently toward opposite sides of the cell by spindle fibers, tiny structures that guide the movement with calm precision. This process ensures that each new cell receives an identical set of chromosomes, preserving the organism's genetic continuity. Chromosome number varies across species. Some plants have hundreds of chromosomes, while others have only a few. Certain animals have unusual arrangements, shaped by their evolutionary histories. Despite this diversity, the underlying principles remain the same. chromosomes act as stable containers for DNA, organizing it in ways that support life's quiet routines. Within each chromosome lie regions called genes, but there are also vast stretches of non-coding DNA. These regions may regulate gene activity, support structural integrity, or reflect remnants of ancient genetic events. They contribute to the chromosome's overall form, shaping how it behaves during replication, repair, and expression. At the tips of chromosomes are telomeres, protective caps that prevent the ends from fraying or merging. Telomeres shorten gradually as cells divide, reflecting the natural passage of time. Their presence helps maintain the chromosome stability, allowing the cell to undergo many cycles of growth before reaching its limits. In certain cells, such as stem cells and reproductive cells, telomerase helps restore telomere length, giving these cells the ability to divide over longer periods. Another important aspect of chromosomes is their unique positioning within the nucleus. They do not float randomly. Instead, each chromosome occupies a specific region known as a chromosome territory. These territories help reduce interference between chromosomes and support efficient gene expression. The quiet spatial arrangement reflects an underlying order, a map that the cell follows without conscious direction. Some chromosomes carry specialized regions called centromeres and telomeres, while others contain nucleolar organizers, sequences that guide the formation of the nucleolus. The nucleolus is where ribosomes begin to take shape, making it an essential center of protein synthesis. The positioning of these regions within the nucleus influences how the chromosome behaves during the cell cycle. Occasionally, chromosomes undergo changes such as inversions, translocations, or duplications. These structural variations reflect the natural fluidity of the genome. Some changes have no noticeable effect, while others may influence traits or contribute to genetic diversity within a population. Across evolutionary time, chromosome structure has shifted quietly, adapting to the needs of different organisms. In certain species, chromosomes behave differently depending on the organism's sex. Humans have two sex chromosomes, X and Y. Females have two X chromosomes, while males have one X and one Y. The Y chromosome carries genes related to male development, but it is much smaller than the X chromosome. To balance gene expression between males and females, one X chromosome in female cells becomes mostly inactive. This inactivation occurs early in development and forms a structure known as a Barr body, a quiet, condensed mass of DNA that remains largely silent throughout life. Despite their complexity, chromosomes remain stable. Their structure protects the DNA, coordinates its distribution, and ensures that each generation receives the essential information needed for life. The calm architecture of chromosomes allows the genome to function with both precision and flexibility, supporting the countless processes unfolding within each cell. Beyond the genes that code for proteins lies an even larger world of sequences that guide when, where, and how those genes are expressed. These regions, known collectively as regulatory DNA, act like gentle conductors of the genome's intricate symphony. They do not produce proteins themselves. Instead, They shape the rhythm and pattern of gene activity, helping cells maintain balance and respond to their environment with calm precision. Regulatory DNA includes promoters, enhancers, silencers, and insulators. Each plays a distinct role, but all contribute to the quiet coordination of genetic expression. Promoters sit just upstream of genes, marking the place where transcription begins. They provide a soft invitation for RNA polymerase to attach and start reading the instructions. Without promoters, the cell would have no clear starting point for transcription, and the gentle flow of information from DNA to RNA would lose its structure. Enhancers, on the other hand, can reside far from the genes they regulate. They act as distant signals, looping through the three-dimensional space of the nucleus to make contact with their target genes. When an enhancer is active, it increases the likelihood that its associated gene will be expressed. This influence is subtle but powerful. Enhancers help fine-tune gene activity, ensuring that proteins are made at the right time, in the right place, and in the right amount. Silencers work in a similar manner, but produce the opposite effect. When active, they reduce or suppress gene expression, allowing the cell to maintain calm control. Silencers prevent unnecessary activity, conserving energy and maintaining order within the genome. Their influence helps keep certain genes quiet until they are truly needed. Insulators are another layer of regulation. They act as boundaries between regions of DNA, preventing enhancers or silencers from influencing genes outside their intended areas. These boundaries help maintain the structure of chromatin, creating gentle zones of activity and rest. Insulators keep the genome organized, ensuring that regulatory signals remain clear and precise. Together, these elements create a nuanced regulatory landscape that extends far beyond the protein coding regions. In fact, only a small percentage of the genome actually codes for proteins, while regulatory sequences make up a significant portion of the remaining DNA. This large non-coding component was once considered inactive, but we now understand that it plays a vital role in shaping life's internal rhythms. Regulatory DNA also interacts closely with transcription factors, proteins that recognize specific sequences and influence gene expression. Some transcription factors activate genes, while others repress them. They bind to regulatory regions with soft precision, responding to signals from within the cell or cues from the environment. This interaction creates a flexible system in which gene expression can rise or fall as conditions change. Another layer of regulation occurs through chromatin remodeling. Proteins responsible for adjusting the structure of chromatin help open or close sections of DNA, influencing which genes can be accessed. When chromatin is tightly packed, the underlying DNA remains silent. When it relaxes, genes in that region become more available. This dynamic structure allows the genome to respond smoothly to developmental stages, environmental conditions, and internal signals. Regulatory DNA also plays a role in long-distance communication within the genome. Some regions form loops that bring distant parts of the DNA into close proximity. These loops help coordinate gene networks, linking genes that need to be co-expressed or silenced together. The formation and maintenance of these loops are guided by proteins and structural elements that shape the broader architecture of the genome. In developing organisms, regulatory sequences help ensure that genes activate in the correct order. During early development, genes responsible for basic structure and body plan are expressed first. Later, genes involved in refinement and specialization begin to activate. This sequence unfolds gradually, guided by the coordinated work of promoters, enhancers, and other regulatory elements. Environmental influences also leave gentle imprints on regulatory DNA. Stress, nutrition, and physical activity can lead to adjustments in regulatory patterns, shaping how genes behave over time. These changes do not alter the underlying DNA sequence. Instead, they influence the way the genome interprets its instructions, creating subtle shifts in gene activity that reflect life's experiences. Regulatory DNA helps maintain the identity of different cell types. In a neuron, enhancer regions that support neural function remain open and active, while regions related to muscle development stay closed. In a muscle cell, the pattern reverses. These stable regulatory landscapes ensure that each cell type maintains its proper identity, even though all cells contain the same genetic blueprint. Throughout the genome, regulatory DNA provides the soft, underlying control that keeps genetic expression balanced. It ensures that essential genes activate at the right times, that unnecessary genes remain silent, and that each cell maintains harmony within its internal environment. Even though DNA carries the core instructions for life, another quiet layer rests above the sequence, a layer known as epigenetics. Epigenetics does not change the letters of the genetic code. Instead, it influences how those letters are read, shaping the cell's behavior in soft, reversible ways. This additional layer provides flexibility, allowing organisms to respond gently to their environment while maintaining the stability of their DNA. One of the most well-known epigenetic mechanisms is DNA methylation. This process involves the addition of small chemical groups called methyl groups to specific bases within the DNA. These groups act like subtle marks that influence gene expression. When a region of DNA becomes heavily methylated, it often becomes less active. The gene lies quiet beneath the mark, resting in silence until conditions shift. When methyl groups are removed, the gene may awaken, becoming accessible once more. Histone modification is another form of epigenetic regulation. DNA winds around histones, forming loops and coils that create the structure of chromatin. Histones can be modified in many ways, by adding or removing chemical groups that influence how tightly the DNA wraps around them. These modifications do not alter the underlying sequence. Instead, they adjust the openness of the chromatin, guiding which genes are available for expression. Tightly packed regions remain silent, while relaxed regions become active and accessible. Epigenetic marks accumulate gradually, shaped by a person's experiences, environment, diet, and even patterns of sleep and stress. Over time, these marks create a gentle record of life's interactions with the genome. Some marks fade quickly, while others persist for long periods. In certain cases, Epigenetic patterns can even be passed from one generation to the next, forming a soft connection between ancestral environments and present-day gene expression. During early development, epigenetics plays an essential role in shaping cell identity. As cells differentiate, they adopt epigenetic patterns that support their specific functions. Neurons develop marks that facilitate communication and signaling. Muscle cells develop patterns that support contraction and strength. Skin cells acquire patterns that maintain protection and renewal. These epigenetic landscapes help lock in the identity of each cell type, ensuring stability throughout life. Environmental factors can gently shift epigenetic patterns. Nutrition influences which enzymes are available to add or remove epigenetic marks. Physical activity can adjust the openness of chromatin in certain tissues. Stress and relaxation create their own subtle signals, guiding the genome's interpretation of its instructions. Even small lifestyle changes can ripple softly through the epigenetic layer, gradually shaping how genes behave. Epigenetics also plays a role in memory and learning. In the brain, certain experiences lead to stable changes in gene expression within neurons. These changes help strengthen connections between cells, supporting the formation of long-term memories. Although the genetic sequence remains the same, epigenetic marks create gentle shifts that allow the brain to adapt to the patterns of our lives. Another important epigenetic phenomenon is genomic imprinting. In some genes, only one copy, either the maternal or the paternal, is expressed, while the other remains silent. This selective expression is controlled by epigenetic marks that distinguish the parental origin of the gene. Imprinting ensures that certain developmental signals follow a balanced pattern, contributing to the calm progression of growth. Epigenetic processes also help regulate the timing of developmental milestones. They guide the transition from one life stage to another, ensuring that genes activate or deactivate in the correct sequence. This coordination supports the gentle unfolding of childhood, adolescence, adulthood, and aging, maintaining order and stability within the body. In aging, epigenetic patterns gradually change. Regions that were once tightly silenced may become more open, while previously active regions may quiet down. These shifts reflect the natural passage of time, creating subtle signatures that accumulate across years. Researchers sometimes refer to this pattern as the epigenetic clock, a gentle indicator of biological age. Despite their influence, epigenetic marks are flexible. Cells have mechanisms to add, remove, and adjust these marks as needed. This flexibility gives the organism resilience, allowing it to adapt to challenges while maintaining overall stability. The genome provides the foundation, but epigenetics allows the organism to shape its responses to the world. Across all life forms, epigenetics adds a layer of nuance to genetic regulation. It provides depth to the genome structure, allowing identical sequences to behave differently depending on context. The gentle marks that shape gene expression illustrate how dynamic and responsive living systems can be. Genetic inheritance is the quiet thread that connects one generation to the next. Every person carries a blend of DNA passed down from their parents, who received their DNA from the parents before them, forming a long, unbroken chain reaching back through time. This continuity allows life to retain its patterns while still welcoming gentle variation. Inheritance begins with the formation of reproductive cells, eggs in females and sperm in males. These cells contain only one set of chromosomes rather than the usual two. The reduction occurs through a calm, elegant process called meiosis, in which a single cell divides twice to produce 4 cells, each with half the original number of chromosomes. This ensures that when an egg and sperm merge, the resulting embryo receives the correct number of chromosomes. During meiosis, chromosomes pair up and exchange small segments of DNA in a process known as crossing over. This exchange happens gently, guided by precise molecular interactions that allow the chromosomes to swap equivalent sections. The result is a blend of genetic information, creating new combinations of traits that did not exist in either parent alone. This recombination is one of the primary sources of genetic diversity in populations. Each pair of chromosomes also separates independently during meiosis, meaning the combination of maternal and paternal chromosomes that end up in each reproductive cell occurs by chance. This independent assortment adds another layer of variation, ensuring that each egg or sperm carries a unique mix of traits. Even siblings with the same parents receive different combinations of their ancestors' DNA. When fertilization occurs, the genetic material from the egg and sperm merges to form a complete set of chromosomes. Half of the DNA comes from one parent, half from the other. This blending creates the foundation of individuality, a new arrangement of genetic instructions shaped by generations of inheritance and the quiet randomness of meiosis. Some traits follow simple patterns of inheritance. For example, certain characteristics are influenced by dominant and recessive alleles. A dominant allele expresses its trait even when paired with a different version, while a recessive allele expresses its trait only when both copies are the same. These patterns were described long ago through gentle experiments with plants, revealing some of the earliest insights into genetic behavior. Other traits follow more complex patterns. Some are influenced by many genes working together, each contributing a small effect. Height, skin tone, and many aspects of personality arise from the combined influence of numerous genetic and environmental factors. These traits do not show clear-cut inheritance patterns. Instead, they display a spectrum of variation. There are also traits linked to the sex chromosomes. Genes on the X chromosome can behave differently depending on whether they appear in males or females. Since males have only one X chromosome, a recessive trait on that chromosome may express itself more readily. Females with 2 X chromosomes have a backup copy that may soften the effect. These patterns add another layer of complexity to inheritance. Mitochondrial DNA follows a different path. Unlike nuclear DNA, which comes from both parents, mitochondrial DNA is inherited almost exclusively from the mother. This maternal inheritance forms a quiet genetic line that stretches back through countless generations of women. researchers often use mitochondrial DNA to trace ancient migrations and understand human ancestry. Inherited traits can reflect both stability and change. Many characteristics remain consistent across generations, carried faithfully in the genome. Others shift gradually, influenced by mutations, recombination, and environmental interactions. The combination of stability and variation allows populations to remain cohesive while adapting softly to new conditions. Some inherited conditions arise when a gene carries a harmful mutation. In many cases, the effects are softened by the presence of a normal copy of the gene. Only when both copies carry the mutation do the effects become more apparent. This recessive pattern helps explain why certain genetic conditions can remain hidden within families for many generations. In other cases, a single altered copy of a gene may influence traits through a dominant inheritance pattern. These traits can appear in every generation, reflecting the strength of the allele's influence. Yet even here, the expression can vary among individuals, reminding us that inheritance is shaped by many subtle factors. The rhythm of inheritance is ongoing. Each generation receives a carefully packaged set of chromosomes, enriched by centuries of history and softened by the quiet processes of meiotic recombination. These small variations accumulate gently over time, creating the diversity observed across human populations. Inheritance also connects us to our evolutionary past. The patterns in our DNA reveal stories of ancient migrations, adaptations to different climates, and the emergence of traits that allowed our ancestors to thrive. Even though these events occurred long ago, their echoes remain within our chromosomes, carried silently from one life to another. Across all of biology, inheritance provides continuity. It ensures that organisms resemble their parents while still possessing their own unique identities. The quiet stability of genetic transmission allows life to persist through time, adapting slowly to new challenges while maintaining its essential patterns. DNA has existed for billions of years, carrying the quiet history of life from its earliest beginnings to the present day. Over these immense stretches of time, the molecule has remained remarkably consistent. The same gentle double helix that guides human development also guided ancient organisms that lived long before the first animals appeared. Through countless generations, DNA has served as the central thread that connects all living things. In the earliest stages of life on Earth, primitive cells carried genetic material that resembled modern DNA, though it may have been simpler. These early molecules helped guide the growth and replication of the first living structures. Over time, DNA evolved to become more stable and efficient, adopting the familiar double-stranded form that allows for accurate copying and reliable transmission across generations. As life diversified, DNA accumulated small changes that allowed organisms to adapt to new environments. Some sequences expanded, others contracted, and new combinations emerged. These gradual changes shaped the diversity of life, the rise of plants, the appearance of animals, and the development of complex organisms. Each shift reflected a quiet adjustment within the genome, allowing life to refine its structures and behaviors. One of the most profound evolutionary innovations involved the development of eukaryotic cells, cells with nuclei that protect and organize DNA. This shift allowed for greater complexity, enabling the emergence of larger organisms with specialized tissues. The genomes of these cells became more elaborate, containing not only protein-coding genes, but also regulatory sequences that guided sophisticated patterns of gene expression. Another turning point occurred when certain cells developed the ability to form multicellular organisms. Instead of living as single units, these cells cooperated, sharing genetic instructions that allowed them to function as tissues, organs, and entire bodies. DNA facilitated this cooperation by providing consistent instructions while allowing flexibility in gene expression. This balance supported the emergence of the first simple animals and, eventually, the vast variety of life on Earth today. Throughout evolutionary history, DNA has interacted with countless influences: environmental changes, mutations, and natural selection. Some changes provided advantages, helping organisms survive and reproduce. Others were neutral, drifting through populations without significant effect. Over millions of years, these small variations accumulated, shaping the genetic landscapes of species. Horizontal gene transfer also played a role in evolution, particularly among bacteria. Unlike animals and plants, which pass DNA through reproduction, bacteria can acquire genetic material from their environment or from other organisms. This ability allows them to adapt quickly, gaining new traits such as antibiotic resistance or metabolic capabilities. These exchanges contribute to the rapid evolution observed in microbial communities. Viruses have also influenced DNA over time. Some viral sequences become integrated into the genomes of their hosts, remaining there for generations. These viral remnants, known as endogenous retroviruses, make up a surprising portion of many genomes. While most remain inactive, some have been co-opted by organisms for beneficial purposes, such as guiding early developmental processes. Their presence reflects the long and complex interactions between viruses and living cells. Across species, certain genes have remained remarkably conserved. These genes perform essential functions, such as building proteins involved in metabolism, cell division, or structural stability. Their sequences change very slowly, reflecting the importance of their roles. When scientists compare the DNA of different organisms, they often find these conserved genes appearing again and again, highlighting the shared ancestry of life. Other genes evolve more quickly, adapting to the needs of specific environments. These genes may influence traits such as immune responses, sensory perception, or physical adaptations. Their faster evolution provides the flexibility needed for organisms to survive in changing conditions. The balance between stability and change allows life to maintain continuity while exploring new possibilities. Mitochondrial DNA offers a unique perspective on evolution. Because it is inherited almost exclusively from the mother and changes at a steady rate, mitochondrial DNA serves as a quiet molecular clock. Researchers use this clock to estimate when populations diverged, trace ancient human migrations, and uncover patterns in the evolutionary history of species. Over time, entire genomes can expand and contract. Some species accumulate large amounts of repetitive DNA, while others maintain compact genomes. These differences arise from duplications, deletions, and the activities of transposable elements, segments of DNA that can move to new locations within the genome. Transposable elements contribute to genetic diversity and evolution, adding gentle fluidity to the structure of DNA. Despite the vast diversity in genome size and structure across species, the fundamental properties of DNA remain unchanged. The molecule continues to store information, replicate with accuracy, and support the quiet processes that sustain life. Its resilience reflects an elegant design shaped by billions of years of evolution. The story of DNA is the story of life itself, a tale of continuity, adaptation, and gentle transformation. Each change, whether small or large, contributes to the tapestry of biological diversity observed today. Through all these shifts, DNA remains a steady presence, guiding organisms through time with silent precision. DNA carries more than instructions for building and maintaining the body. It also preserves a gentle record of ancestry, a quiet biological memory that stretches across countless generations. Within each cell, patterns in the genome reflect ancient migrations, shared lineages, and the long journey of life on Earth. These patterns do not speak loudly. They rest silently within the sequence, waiting to be uncovered and interpreted. Ancestral signals appear in many forms. Some reside as in the small variations that have drifted through populations over thousands of years. Others linger in conserved regions that have remained unchanged for ancient reasons. Together, these features create a soft map of origins, linking individuals to broad branches of the human family tree. One of the clearest markers of ancestry is found in mitochondrial DNA. Because it is inherited almost entirely from the mother, mitochondrial DNA forms a continuous maternal line that traces back through time. Each generation adds a few gentle variations, creating distinct clusters known as haplogroups. These haplogroups reflect ancient migrations, movements of early humans across continents and into new environments. Scientists can follow these subtle patterns to understand how populations expanded and settled around the world. Another quiet source of ancestral information comes from the Y chromosome, which passes from father to son. Like mitochondrial DNA, the Y chromosome accumulates changes gradually, forming its own haplogroups. These paternal lineages offer another view of human history, revealing migration routes, population splits, and long-standing connections among groups. While only males carry the Y chromosome, its patterns contribute to a broader understanding of ancestry. Ancestral information also emerges from autosomal DNA, the 22 pairs of non-sex chromosomes that everyone carries. Unlike mitochondrial DNA or the Y chromosome, autosomal DNA recombines with each generation, blending together contributions from many ancestors. This blending softens the boundaries of lineage, creating a mosaic that reflects a wide network of relatives across centuries. Even though recombination mixes the sequences, some regions remain linked. forming blocks of DNA passed intact through multiple generations. These blocks allow researchers to identify shared ancestry between individuals. If two people carry similar segments of DNA, it suggests they share a common ancestor somewhere in their family histories. The length and number of these shared segments help estimate how closely they are related. In this way, the genome becomes a quiet record of family connections, holding information that stretches far beyond living memory. Certain genetic variations, known as ancestry-informative markers, are more common in some populations than others. These markers do not determine identity or culture. Instead, they reflect historical patterns, the environments, migrations, and events that shaped different regions of the world. The presence of these markers in an individual's DNA offers a soft indication of ancestral origins, revealing which parts of the world contributed to their genetic makeup. Even ancient events leave traces in the genome. Thousands of years ago, early humans encountered other hominin groups, such as Neanderthals and Denisovans. Small amounts of DNA from these groups remain in modern populations, preserved through quiet interbreeding during ancient migrations. These traces do not define individuals, but they offer insight into moments when different human groups crossed paths and shared their genetic heritage. Ancestry signals also appear in patterns of genetic diversity. Some populations carry more variation because they represent older lineages with longer histories of accumulated changes. Others show less variation, reflecting population bottlenecks or periods of isolation in their past. These patterns reveal how different groups expanded, contracted, or adapted to the challenges of their environments. The genome also holds memories of adaptation. Certain variations became more common because they helped ancestors survive specific conditions. For example, some variants support life at high altitudes. Others influence how the body processes nutrients or responds to sunlight. These adaptations do not arise quickly. They accumulate slowly, woven over generations into the quiet fabric of the genome. Even though ancestry signals are subtle, they form part of the broader story written in DNA. They reflect the journeys of countless individuals whose lives shape the genetic landscape we inherit today. The genome is not a fixed record. It evolves gently as populations shift, cultures change, and environments reshape the challenges of survival. Within each person, these ancestral patterns blend seamlessly. No single region of DNA defines who someone is or where they come from. Instead, ancestry is a mixture, a soft combination of past influences and present individuality. The genome carries the echoes of ancient movements, but it also reflects millions of unique combinations that make every person distinct. Ancestral information exists quietly within the genome, but it can be uncovered through careful study. Scientists compare DNA sequences from individuals around the world, looking for patterns that reveal connections across time and geography. These comparisons help reconstruct the deep history of humanity, tracing how early groups spread from their origins and adapted to new environments across all cultures and populations. The genome tells a story of shared beginnings. Despite differences in appearance, language, or tradition, humans are deeply connected through the patterns of their DNA. The quiet memory of ancestry reminds us that the diversity observed today arises from a long and interconnected past. Even though DNA often feels like a distant concept, something tucked away in the nucleus, resting silently within the cell, it plays a constant role in everyday life. Every moment, countless processes depend on the steady instructions held within the genome. These instructions shape how cells behave, how tissues maintain themselves, and how the body responds softly to the shifting demands of the environment. In each cell, DNA guides the creation of proteins, which carry out most of the body's essential functions. Some proteins act as enzymes, facilitating gentle chemical reactions that keep metabolism flowing. Others become structural elements, giving cells their shape and strength. Still others form channels that let ions drift in and out of cells, supporting calm electrical signals in the brain and steady rhythms in the heart. All of these activities trace back to the instructions encoded quietly within the DNA. DNA also helps cells maintain their identity. A skin cell reads only the genes that support protection and renewal. A liver cell activates genes that manage detoxification and metabolism. A neuron expresses genes that help with communication and signal transmission. Even though all cells carry the same genome, each interprets it differently, creating the diversity of functions required for complex life. In tissues that regularly renew themselves, such as skin, blood, and the lining of the digestive system, DNA guides the cycle of growth and replacement. Stem cells in these tissues divide gently, producing new cells that take on specific roles. The instructions for this process are written into the DNA, but the expression of those instructions shifts over time, adjusting to the body's needs. This quiet renewal allows organs to remain healthy and functional throughout life. The immune system also relies on DNA to create diversity. Immune cells generate a wide range of receptors that help recognize potential threats. To produce this diversity, certain regions of immune cell DNA rearrange themselves during development. These rearrangements are precise but flexible, creating countless variations that enable the body to identify unfamiliar molecules. The result is a gentle but powerful system that protects the body from harmful organisms while remaining adaptable. DNA influences how the body responds to stress. When a cell encounters heat, cold, or chemical imbalances, it activates specific genes that help stabilize its internal environment. These genes produce proteins that repair damage, adjust metabolic pathways, or create temporary protective structures. The activation of stress response genes remains measured and controlled, helping the body maintain balance during challenging moments. Hormones also interact with DNA. When hormones enter cells, they bind to receptors that travel to the nucleus. These receptors attach to specific regulatory regions of DNA, encouraging or discouraging the expression of certain genes. This gentle signaling allows tissues to coordinate their functions. For example, hormones help regulate growth, metabolism, mood, and reproductive cycles, all through quiet interactions with the genome. In the brain, DNA influences learning and memory. When a person repeatedly experiences something, a sound, a movement, an emotion, neurons strengthen certain connections. Part of this strengthening depends on gene expression. Neurons activate genes that produce proteins involved in creating or reinforcing synapses. Although the DNA sequence does not change, the patterns of activation shift softly in response to experience. Over time, these shifts contribute to the formation of long-term memories. DNA also plays a role in circadian rhythms. Many cells contain internal clocks that cycle over roughly 24 hours. These clocks depend on genes that activate and repress themselves in a rhythmic pattern. The gentle oscillation of these genes helps regulate sleep, alertness, hormone levels, body temperature, and metabolic activity. Even without external cues, the genome maintains this internal rhythm, guiding the body through daily cycles. In muscles, DNA helps regulate growth and repair. When muscles experience small amounts of stress, through exercise or everyday movement, certain genes activate to strengthen the tissue. These genes guide the production of proteins that rebuild muscle fibers, enhance energy production, and support endurance. The process unfolds gradually, guided by a balance of genetic signals and biochemical responses. DNA influences the senses as well. Genes encode the proteins that detect light in the eyes, vibrations in the ears, chemicals in the nose and mouth, and pressure on the skin. These sensory proteins allow the body to interpret its surroundings, creating the quiet continuity of perception that supports everyday life. In metabolism, DNA provides the instructions for breaking down nutrients and converting them into usable energy. Enzymes that process carbohydrates, fats, and proteins are all encoded in the genome. These pathways operate constantly, supporting the brain, muscles, and organs. The smooth coordination of these reactions reflects the underlying stability of genetic instructions. Throughout life, DNA remains the steady foundation of the body's functioning. It does not dictate every action directly, but it provides the framework that supports all cellular behavior. Its instructions are read in patterns, sometimes active, sometimes resting, depending on the needs of the moment. Even though DNA is hidden deep within the nucleus, it shapes the quiet routines of the body from waking to sleeping, from thinking to breathing. It guides development, supports maintenance, and allows the organism to respond gently to the world. The calm presence of DNA in every cell reflects the stability that life depends on. A silent, enduring structure that works continuously behind the scenes. Thank you for joining this quiet exploration of DNA. From the delicate structure of the double helix to the soft patterns of inheritance, every part of this journey reflects the calm, steady rhythm that carries life forward. Even the smallest molecules inside our cells move with patience, helping the body grow, adapt, and endure with quiet strength. As you rest now, you can let all of these ideas drift away. There is nothing you need to remember, nothing you need to hold on to. Just the gentle sense of having wandered through something peaceful, something steady, something ancient and comforting. If you're still awake, you might notice your breathing becoming slower, your muscles softening, your thoughts settling into a calm, open space. And if you've already drifted off, that's completely all right. This space is here for you, to help you unwind, to ease your mind, and to give you a place where curiosity and relaxation can coexist softly and quietly. Thank you for spending this time here, and until next time, I hope you have a deep, restful sleep. Good night.